Gene therapy is an experimental technique that uses DNA to treat or prevent disease. The first FDA-approved gene therapy experiment in the United States occurred in 1990, when Ashanti Da Silva was treated for ADA. ADA is adenosine deaminase deficiency, a rare immune system deficiency that is caused by a lack of an enzyme, adenosine deaminase. Those that suffer from ADA are susceptible and defenseless against numerous diseases and infections, and oftentimes they die before their first birthday. Since then, over 1,700 clinical trials have been conducted using a number of techniques for gene therapy. In gene therapy, DNA that encodes a therapeutic protein is packaged within a vector which is used to get DNA inside cells within the body. A vector is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell, where it can be replicated and or expressed. Once inside, the DNA becomes expressed by the cell machinery, resulting in the production of therapeutic protein, which in turn treats the patient's disease. Although early clinical failures led many to dismiss gene therapy as overhyped concept, yet the clinical success in the recent years have induced new optimism in the promise to make gene therapy a successful approach to treat disease. This includes successful treatment of patients with the retinal disease, X-linked SCID, ADA SCID, adrenoleukodystrophy, and Parkinson's disease. Recent breakthrough using gene therapy is a mouthwash that promises good results for those with oral cancer. This mouthwash wards off oral cancer by destroying suspicious growth in the mouth before they become malignant. Types of gene therapy. First, somatic gene therapy. In somatic gene therapy, the therapeutic genes are transferred into the somatic cells or body of a patient. 2. Germline gene therapy. In germline gene therapy, germ cells, sperm or ovum are modified by the introduction of functional genes, which are integrated into their genomes. This would allow the therapy to be heritable and passed on to later generations. And the last one is preventive gene therapy. Preventive gene therapy is the repair of a gene with a mutation associated with a progressive disease prior to the expression of a medical condition in order to prevent that expression. Advantages of gene therapy. First, Gene silencing is a concept that in itself is self-efficient for management of many diseases. 2. Gene therapy has the potential to eliminate and prevent hereditary disease such as cystic fibrosis and is a possible cure for heart disease, AIDS, and cancer. And the last one gives an advantage to a person born with genetic disorder to live life in a normal way by replacing non-functional gene with a functional one.
Gene therapy is the introduction of genes into existing cells to prevent or cure a wide range of diseases. For example, suppose a brain tumor is forming by repeatedly dividing cancer cells. The other target of some of the various retrovirus used include bone marrow, skin, and liver cells.
Gene therapy today is approaching the faulty genes that can cause genetic diseases that inherited through generations. The process involves using immune cells that have the ability to develop into the faulty cells that have different functions. However, the idea of the gene therapy can be controversial because though it can prevent the family from having some genetic disorder, it also might affect the fetus development in unexpected ways. So it still remains debatable till today whether it's actually good for human, and some also suspect its longevity as there isn't enough background studies to support. To put in personal opinions, gene therapy can be pretty similar to the organ transplantation in the sense that it allows bad genes to be replaced by stronger ones. And it's noticeable that most arguments are coming from the same place, which is the ethical concerns, because people are worried that the scientists will mess around with the genetic creatures and start creating human clones or whatever, what can be worse. And also another factor that may hinder most people from getting the gene therapy is its cost. It is estimated that the therapy costs approximately up to $4 million per person in the US. So it's hard to justify when we have such technology to create cures for some of the world's most serious diseases, but only accessible to a certain group of people.